Hey everyone, welcome to another deep dive with us.
It's great to be here.
Now today we're going to be tackling a topic that I hear is front and center in Mr. Martin's IB bio class right now.
Oh, yeah.
That's right, we're diving into the world of cells, specifically cell structure.
A classic.
We've got a whole stack of resources that Mr. Martin's recommended to kind of guide us through this deep dive.
Just fantastic.
So yeah, let's jump right in. Now it never ceases to amaze me that like all life forms, from the tiniest bacteria to like giant redwoods, are all built from these fundamental units called cells.
Yeah, it's one of the most remarkable unifying concepts in biology. That unity and diversity. We see all these different life forms, yet they're all built from the same fundamental building block.
That's right. And even though they share that basic building block, they come in such a crazy variety of types and structures, each with their own unique function, right?
Oh, absolutely. It's like saying all houses are made of bricks, but some are cozy cottages and others are skyscrapers, right?
Yeah.
Each with its own unique architecture and purpose.
Exactly. Now, before we get into like all the variations in those house designs, I'm kind of curious about how we even discovered cells in the first place. Like they're microscopic after all. How did we figure that out?
Well, we owe a lot to the development of microscopes.
Of course.
Back in the 1600s, scientists like Robert Hooke started using those simple light microscopes.
Right.
And they were able to observe cells for the first time. Light microscopes, like the ones you probably use in class, they use visible light to magnify objects, and they're still incredibly valuable tools today.
But I'm guessing they have their limitations, right?
Exactly.
Yeah.
And that's where electron microscopes really revolutionized our understanding of cells. Instead of light, they use beams of electrons, which have a much shorter wavelength, and that allows us to see incredibly tiny structures within cells.
Wow.
Things like the intricate folds of membranes, or even those tiny ribosomes that make proteins.
Wow.
It's really amazing. Imagine being able to see the individual gears and springs inside of a watch. That's the level of detail we can achieve with electron microscopes.
So armed with these super powerful tools, scientists were able to figure out that there are two main types of cells, right?
Yes.
Prokaryotes and eukaryotes.
Right.
What is the fundamental difference between these two?
Think of it this way. Prokaryotic cells are like the studio apartments of the cell world.
Okay.
They're really efficient, very compact.
Right.
They lack a true nucleus, and they don't have all those specialized membrane-bound organelles that you find in eukaryotic cells.
Right.
So bacteria and archaea are examples of prokaryotes, and they're incredibly successful life forms.
Yeah.
And thriving on Earth for billions of years.
So where's their DNA if they don't have a nucleus?
Their DNA, it's a single circular chromosome, and it just kind of hangs out in the cytoplasm.
Okay.
In a region called the nucleoid.
Okay.
They also have smaller ribosomes compared to eukaryotes.
Got it. Okay, so then eukaryotic cells are more like what?
They're more like the sprawling mansions of the cell world.
Okay, all right.
So eukaryotic cells have a true nucleus to house their DNA, which is organized into multiple linear chromosomes. And then they have this amazing array of specialized membrane-bound organelles, each with its own unique function.
Right.
It's like having separate rooms for cooking, sleeping, and working out,
I see.
Everything's compartmentalized for efficiency.
Yeah, so like plant cells and animal cells, even those weird protists we were studying, are all eukaryotes.
Exactly.
Okay.
And here's a question to ponder. How did these complex eukaryotic cells evolve from those simpler prokaryotes? One prevailing theory is endosymbiosis.
Okay.
Imagine one cell engulfing another, but instead of digesting it, the engulfed cell became a permanent resident.
Right.
And eventually evolved into organelles like mitochondria and chloroplasts.
So one cell basically ate another and they just became best buddies.
Yeah, in a way. And it's supported by evidence.
How could that even happen?
Well, think about mitochondria. The powerhouses of eukaryotic cells, they have their own DNA. And ribosomes, and they're remarkably similar to those found in bacteria.
Wow.
So that suggests that they were once free-living prokaryotes that formed a symbiotic relationship with a host cell.
That is wild.
Yeah.
So let's explore these eukaryotic mansions in a little more detail. What kind of organelles are we talking about here?
Well, let's start with the control center, the nucleus.
Okay.
It houses the cell's DNA, which is the blueprint for building and running the whole cellular operation.
Like the cell's brain.
Yeah, a good analogy. And it's not just a jumbled mess of DNA.
Right.
The nucleus has a double membrane
Okay.
with these nuclear pores that regulate what goes in and out, like tiny border checkpoints.
So what else is going on in these cells? What other organelles are working hard to keep things running smoothly?
Well, you've got ribosomes, which are like tiny protein factories. They take the instructions from the DNA and translate them into proteins,
Right.
which are the workhorses of the cell,
Yeah.
carrying out all sorts of essential functions.
So if the DNA is like the blueprint, the ribosomes are like the construction workers making all the different parts of the cell.
That's a great way to put it. Now, some ribosomes float freely in the cytoplasm,
Right.
building proteins that the cell needs right there.
Fuck.
But others are attached to a network of membranes called the endoplasmic reticulum, or ER for short.
Okay, and what's happening in the ER? It sounds important.
The ER is like the cell's transportation and manufacturing hub.
Okay,
Think of it as a highway system with factories along the way.
I see.
We've got the rough ER, which is studded with those ribosomes, making proteins that need to be shipped to other parts of the cell or even secreted outside the cell.
And what about the smooth ER? What's that one doing?
The smooth ER is more like a specialized workshop. It focuses on tasks like lipid synthesis and detoxification.
Okay.
It's smooth because it lacks those ribosomes.
So the ER makes proteins and lipids. Where do they go from there?
That's where the Golgi apparatus comes in.
Okay.
It's like the cell's post office. It carefully processes packages and ships those proteins and lipids to their final destinations.
It sounds like there's a lot of compartmentalization and teamwork going on in these eukaryotic cells.
Absolutely.
Each organelle has its own specialized role to play.
Right. And speaking of specialized roles, let's talk about the mitochondria.
Okay.
Those powerhouses of the cell,
Right.
they're responsible for producing ATP.
Yep.
The energy currency of the cell through cellular
I
respiration.
remember learning about that. That's how cells break down glucose to release energy, right?
Exactly.
Yeah.
And their structure is perfectly suited for this task. Mitochondria have a double membrane,
Okay.
with the inner membrane folded into structures called cristae.
Okay.
Think of it like having a wrinkled surface.
Okay.
This increases the surface area available for energy production.
It's all about maximizing efficiency.
Precisely. Now, what about waste management? Cells produce waste just like any other system,
Right.
and they need a way to deal with it. That's where lysosomes come in. They're like the recycling centers of the cell,
Yeah.
breaking down cellular waste and debris.
So they're like tiny garbage disposals.
Yes,
Keeping the cell clean and tidy.
exactly. They contain powerful enzymes that can break down all sorts of molecules, ensuring that the cell stays healthy and functional.
Incredible. And what about storage? Cells need to store nutrients and other essential materials, right?
Right. Again, for that, we have vacuoles. They're like the storage units of the cell, holding water nutrients and even waste products.
Okay.
In plant cells, vacuoles can be quite large, helping to maintain the plant's structure by providing internal pressure.
So plant cells have some unique features too, right?
Yes.
Tell me more about those.
Well, one of the most distinctive features of plant cells is the presence of chloroplasts.
Okay.
The remarkable organelles responsible for photosynthesis.
Yeah.
These are like miniature solar panels,
Right.
capturing light energy from the sun and converting it into chemical energy in the form of glucose, which the plant uses for food.
That's how plants make their own food, right? Amazing.
It is. Chloroplasts also have a double membrane structure,
Okay.
and inside they have stacks of flattened discs called thylakoids, which contain chlorophyll, the green pigment that absorbs light energy.
It sounds like there's a lot going on in those tiny plant cells.
There is.
What else makes them different from animal cells?
Well, besides chloroplasts, plant cells also have a rigid cell wall surrounding the cell membrane. This wall is made primarily of cellulose,
Okay.
and it provides structural support and protection, a bit like a suit of armor.
Oh, so plant cells are tougher than animal cells.
You could say that, yeah.
Interesting. Is there anything else that sets them apart?
Well, those large central vacuoles we talked about earlier.
Right.
They play a key role in maintaining turgor pressure in plants, which helps keep them upright and rigid.
Right.
Think of it like a water balloon providing internal pressure to support the plant.
That makes sense. It's fascinating how plant cells have all these extra features to help them thrive. But we can't forget about fungi.
Right? Oh, that's right.
They're eukaryotes too.
They are. Fungi, like plants, have cell walls, but theirs are made of chitin, a tough material also found in the exoskeletons of insects.
Wow.
And while they don't have chloroplasts, they obtain nutrients by absorbing organic matter from their environment.
So they're like the decomposers of the ecosystem breaking down dead matter.
Exactly. Fungi play a crucial role in nutrient recycling and maintaining a healthy ecosystem.
It's amazing how much diversity there is within the world of eukaryotic cells.
It really is.
They all have their own unique adaptations to survive and thrive.
Absolutely. And don't forget, we've been able to unravel all these intricate details thanks to advances in microscopy.
Yeah.
Being able to identify different cell types and organelles in micrographs is an important skill for IB Bio, so keep practicing.
Mr. Martin definitely stressed that. We've been working on identifying key structures like the nucleus, mitochondria, and Golgi.
It's like solving a cellular puzzle. And remember, it's not just about recognizing those structures.
Right.
The real key is understanding their functions and how they work together to keep the cell alive.
That's where the real learning happens, right? Making those connections between structure and function.
Absolutely.
It's like understanding how all the parts of a complex machine fit together to make it work.
Precisely. But cells aren't just static machines.
Right.
They're constantly interacting with their environment, taking in nutrients, expelling waste, communicating with other cells, and responding to changes in their surroundings.
So it's not just a snapshot. It's a dynamic system constantly adapting and responding.
Exactly. Cells are incredible feats of biological engineering.
Okay, that's amazing. So let's do a side-by-side comparison of those different eukaryotic cells, animal, plant, and fungal, and highlight their key structural differences.
Okay, great idea. Let's start with the most obvious, the presence or absence of a cell wall.
Okay.
Plants have those rigid cellulose walls, fungi have chitin walls, and animal cells lack walls altogether.
So that's a quick and easy way to tell them apart. What about those storage units, the vacuoles?
That's another key difference.
Yeah.
Plants have those large central vacuoles that help maintain turgor pressure,
Right.
while animal cells have smaller temporary vacuoles.
Okay.
And in fungi, vacuole size and function can vary quite a bit.
And of course, we can't forget the energy-producing organelles, chloroplasts, in plants versus mitochondria in all eukaryotes.
All right.
That's a major distinction, reflecting their different ways of obtaining energy.
Right. Plants, as autotrophs, make their own food through photosynthesis, while animals and fungi, as heterotrophs, obtain energy by consuming other organisms or organic matter.
There's also that interesting difference with centrioles, right?
Yes, centrioles play a role in cell division in animal cells, but are absent in plants and most fungi.
So when we're looking at micrographs, we need to be on the lookout for these key structural differences to figure out what type of cell we're seeing.
Exactly. It's all about recognizing those distinctive features and understanding their functional significance.
This has been incredibly helpful. I feel like I'm finally starting to grasp the big picture of cell structure.
That's great to hear.
But remember, this is just the beginning of our exploration.
It is.
There's so much more to discover within the amazing world of cells.
Absolutely.
I'm ready to dive deeper. But for now, maybe we should give our listeners a chance to absorb all this amazing information.
Sounds good.
We'll be back with more insights from our deep dive into cell structure. Welcome back to our cell-tacular exploration. Before we took a moment to process, we were comparing the different types of eukaryotic cells. But let's circle back to prokaryotes for a bit.
Okay.
We touched on the basics, but what are those defining characteristics that really set them apart from eukaryotic cells?
Well, prokaryotes are all about simplicity and efficiency. So, first and foremost, they lack that membrane-bound nucleus that we see in eukaryotes.
Right.
Their DNA is a single circular chromosome, located in a region called the nucleoid, but it's not enclosed within a separate membrane.
So it's just floating in the cytoplasm. That seems so different from our neatly organized eukaryotic cells.
Exactly. Prokaryotes have evolved to do a lot with a little.
Right.
They also lack those membrane-bound organelles, like the mitochondria, the endoplasmic reticulum, and the Golgi apparatus that we find in eukaryotes.
So how do they manage to carry out all the essential functions of life without those specialized compartments? It's like trying to run a whole city without separate buildings for the power plant, the post office, and the factories.
It's remarkable, isn't it?
It is.
They've developed incredibly efficient ways to carry out those functions within their simpler structure. For example, while they lack mitochondria, many prokaryotes can still perform cellular respiration, but they use their cell membrane to generate ATP.
Oh, I see. So it's like they figured out how to generate power without needing a dedicated power plant.
Precisely. They're incredibly adaptable.
Right.
Now let's talk about ribosomes.
Okay.
Both prokaryotes and eukaryotes have ribosomes, those protein synthesis machines. but there's a key difference in their size.
Right. Mr. Martin mentioned that eukaryotes have larger ADS ribosomes, while prokaryotes have smaller 70S ribosomes.
That's right.
Does that size difference have any practical implications?
It does. It actually plays a crucial role in how some antibiotics work.
Oh.
Certain antibiotics specifically target those 70S ribosomes in bacteria, inhibiting protein synthesis and effectively killing the bacteria without harming our own eukaryotic cells.
So it's like a targeted missile system that only hits bacterial cells.
That's a great way to think about it. And it's one of the reasons why antibiotics are so effective against bacterial infections.
That makes sense.
Now, another key feature of prokaryotes is their cell wall.
Okay.
This rigid outer layer provides structural support and protection, and it's composed of peptidoglycan,
Right.
a unique molecule not found in eukaryotes.
That reminds me of our lab work where we use the Gram stain to differentiate between types of bacteria based on their cell wall structure.
Yes, the Gram stain is a valuable tool in microbiology, helping us identify and classify bacteria based on the differences in their cell wall composition.
Right.
Some bacteria even have an extra protective layer called a capsule or glycocalyx surrounding the cell wall. This capsule can help them evade the immune system and make them more resistant to antibiotics.
Oh, so it's like an extra layer of armor.
Exactly. It's a key factor in how some bacteria cause disease.
I see.
Now, let's not forget about those hair-like extensions called pilia.
Right.
These structures can help bacteria attach to surfaces,
Okay.
or even transfer genetic material between themselves through a process called conjugation.
Conjugation. That's how bacteria can share those antibiotic resistance genes, making them even harder to fight.
Precisely. It's one reason why antibiotic resistance is such a growing concern in healthcare.
Right.
And some prokaryotes also have those whip-like structures called flagella, which enable them to move.
So they're like tiny outboard motors propelling the bacteria around.
A great analogy.
Yeah.
Flagella allow bacteria to swim towards nutrients or away from harmful substances, playing a crucial role in their survival and spread.
Prokaryotes are so much more complex than I initially realized.
They are.
They're incredibly adaptable and resourceful.
They're true survivors, having evolved over billions of years to thrive in practically every environment on Earth.
Okay, that's amazing.
Yeah.
So we got a good handle on prokaryotes now.
Yeah.
Let's shift gears back to those eukaryotic mansions and those fascinating organelles that we were talking about.
Okay.
What are some of the specific adaptations that allow them to carry out their specialized functions so effectively?
Let's start with the mitochondrion, that ATP-producing powerhouse.
Okay.
Its structure is beautifully adapted to its function in aerobic respiration. Remember those cristae, the folds in the inner membrane?
Yes.
They're key to maximizing energy production.
Yes, you mentioned that they increase the surface area.
Yeah.
But I'm not entirely clear on how that helps with ATP production.
Think of it this way.
Okay.
More surface area means more space for the electron transport chain and ATP synthase. The molecular machinery that drives the final steps of ATP production.
Okay.
It's like having a larger workspace to get more work done.
So more surface area equals more energy output. Makes sense. What about the space between the membranes, the inner membrane space? Does that serve a specific purpose as well?
Absolutely. That small space is crucial for generating a proton gradient, which acts like a dam holding back a reservoir of potential energy. This gradient is the driving force for ATP synthase, allowing it to produce ATP efficiently.
It's mind-blowing how these tiny structures work together in such a coordinated way.
It is.
It's a symphony of molecular machinery.
It is.
And let's not forget the matrix, the innermost compartment of the mitochondrion.
Right.
That's where the enzymes for the Krebs cycle, another key stage in cellular respiration, are located.
Exactly.
So the mitochondrion is like a highly organized factory with different departments for each stage of ATP production.
That's an excellent analogy. Now let's turn our attention to another remarkable organelle, the chloroplast, the site of photosynthesis in plant cells.
Right. Chloroplasts are like those miniature solar panels we talked about, right? Capturing light energy and converting it into chemical energy.
Yeah, exactly. And just like the mitochondrion, the chloroplast has a double membrane structure, but its internal organization is quite different.
Okay.
Inside the chloroplast, we have those stacks of flattened discs called thylakoids, which contain the green pigment chlorophyll.
Right. Chlorophyll is what absorbs the light energy, right?
That's right. Chlorophyll molecules are organized into photosystems within the thylakoid membranes.
Okay.
These photosystems capture light energy and use it to split water molecules, releasing electrons and ultimately generating ATP.
So the thylakoids are like the light harvesting antenna of the plant cell.
Precisely. And those stacks of thylakoids, called grana, increase the surface area available for light absorption, making photosynthesis more efficient.
It's fascinating how both mitochondria and chloroplasts, these energy-converting organelles, have evolved these highly folded membranes to increase surface area and maximize their output. It's like a recurring theme in cell structure: form follows function.
You've hit the nail on the head. It's a testament to the power of evolution to optimize structure for specific functions.
Right.
Speaking of form and function, let's delve a bit deeper into the nucleus, the control center of the eukaryotic cell.
Okay, we talked about the nucleus earlier, but I'm still curious about those nuclear pores you mentioned.
Right.
How do they regulate what goes in and out of the nucleus?
Those nuclear pores are incredibly important gatekeepers.
Okay.
They control the movement of molecules between the nucleus and the cytoplasm, ensuring that only the right molecules get in and out.
Right.
They're highly selective,
Okay.
allowing some molecules to pass freely while others require specific signals or transport proteins.
So it's not just a passive barrier, it's
No.
actively controlling traffic.
Exactly. The nuclear pores are essential for coordinating gene expression, ensuring that the right messages get from the DNA to the ribosomes to produce the proteins the cell needs at the right time.
It makes sense that the nucleus would need to tightly control the flow of information to keep the cell running smoothly.
Absolutely. And the nuclear membrane itself also plays a key role in cell division, both mitosis and meiosis. During these processes, the nuclear membrane actually breaks down,
Wow.
allowing the chromosomes to be separated and distributed to the daughter cells.
Okay.
Then it reassembles in each new cell.
Wow, so it's not a static structure. It can disassemble and reassemble as needed.
Exactly. It's a dynamic process that highlights the adaptability of cell structures.
This is all so fascinating. It's like peeling back the layers of a complex onion, revealing more and more intricate details with each layer.
I'm glad you're finding it so engaging.
I am.
And remember, we're just scratching the surface of the amazing world of cell structure. There's still so much more to explore.
I can't wait to delve deeper. But for now, maybe we should pause and let our listeners process all this incredible information before we move on to the next exciting aspect of cell structure. Welcome back to the Deep Dive. We've been on quite a journey exploring the intricate world of cell structure. And before our little pause, we were discussing the amazing adaptations of those energy-converting organelles, mitochondria and chloroplasts.
Right.
Now, let's shift our focus to another crucial process: the protein production and transport system within eukaryotic cells.
Ah, yes. Protein synthesis and transport, a fundamental aspect of cell function.
Yeah.
These protein molecules are the real workhorses of the cell, carrying out a vast array of tasks, from catalyzing reactions to transporting molecules across those cellular membranes.
Right. I remember Mr. Martin saying proteins are involved in practically everything the cell does.
They really are.
So where does this protein journey begin?
It all starts with those remarkable protein synthesis machines, ribosomes. They take the genetic code from messenger RNA or mRNA and translate it into polypeptide chains,
Okay.
which then fold into those functional proteins we've been talking about.
And if I recall correctly, there are two types of ribosomes in eukaryotic cells.
Yes.
Those free-floating in the cytoplasm.
Yes.
And those attached to the rough endoplasmic reticulum.
You've got it.
Okay.
Free ribosomes synthesize proteins that are destined to stay within the cytoplasm,
Okay.
while those attached to the rough ER are making proteins that will be transported to other organelles or even secreted outside the cell.
So the rough ER is like a protein processing and packaging plant.
Yeah.
Making sure everything is in order before it gets shipped out.
That's a great analogy. As proteins are synthesized by those ribosomes on the rough ER, they enter the ER lumen, which is the space between the ER membranes.
Okay.
Inside this lumen, the proteins undergo folding modification and a sort of quality control check.
It sounds like a busy workshop in there.
It is.
What kind of modifications are we talking about?
Well, a number of things can happen. For example, glycosylation is the addition of sugar molecules.
Okay.
And disulfide bonds can form to help stabilize the protein's structure.
Right.
These modifications are essential for ensuring that the protein can function correctly.
So it's not just about making the protein.
Right.
It's about making sure it's in tip-top shape before it moves on.
Exactly. The rough ER acts like a quality control inspector, ensuring that only properly folded proteins are allowed to leave and continue their journey.
Right.
Misfolded proteins are flagged and degraded, preventing them from causing problems within the cell.
It's like having a dedicated team of inspectors making sure every product meets the highest standards before it leaves the factory.
Precisely. And once the proteins have passed inspection in the rough ER,
Yeah.
they're packaged into vesicles, small membrane-bound sacs.
Okay.
that bud off from the ER and travel to the Golgi apparatus.
Ah, yes, the Golgi apparatus, the cell's post office.
Exactly.
Sorting and delivering packages all over the cell.
Exactly. The Golgi apparatus is like a series of stacked, flattened membrane sacs or cisternae, okay, each with its own set of enzymes. As proteins move through these different compartments, they undergo further modifications and get sorted based on their final destinations.
It's like a highly organized distribution center with different departments for processing and labeling packages.
A perfect comparison.
Yeah.
Once the proteins reach their designated location within the Golgi, they're packaged into new vesicles, which bud off from the Golgi and are then transported to their final destinations.
Okay.
Some vesicles even fuse with the cell membrane, releasing their contents outside the cell through a process called exocytosis.
So that's how cells secrete hormones, enzymes, and other signaling molecules.
Exactly.
Fascinating.
It's a fundamental process for cell communication and regulation.
This whole system of protein production, modification, and transport is amazingly complex and coordinated.
It is. It's a testament to the incredible organization within eukaryotic cells.
Yeah, and remember, all of this is happening thanks to those membrane-bound organelles.
Right.
Those compartments allow for the separation and specialization of different cellular processes,
Yes.
leading to much greater efficiency and control.
Absolutely.
Right? It's like having specialized departments within a factory, each focusing on a specific task.
Exactly.
Now, since we're talking about specialization, what about the unique adaptations we see in cells that are designed for very specific functions?
Ah, that's a great point. Think about red blood cells or erythrocytes, which are specialized for oxygen transport.
Right.
These cells are incredibly efficient at picking up oxygen in the lungs and delivering it to tissues throughout the body.
Right, like tiny oxygen delivery trucks.
Exactly.
What makes them so good at this job?
Well, for one, their shape is key. They're biconcave, thinner in the center than at the edges.
Okay.
This shape increases their surface area to volume ratio,
Right.
allowing them to absorb and release oxygen more effectively.
So once again, it's all about maximizing surface area for exchange.
Precisely. Red blood cells also lack a nucleus. And most other organelles, leaving more space for hemoglobin, the iron-containing protein that binds to oxygen.
That's amazing. They've essentially streamlined their structure to become super efficient oxygen carriers.
Exactly. Another great example is the proximal convoluted tubule cell in the nephron, the functional unit of the kidney.
Okay.
These cells are responsible for reabsorbing essential nutrients and water from the filtrate, preventing them from being lost in the urine.
So they're like tiny recycling centers in the kidneys.
Yes.
Making sure we don't lose those valuable nutrients.
Precisely. To maximize their reabsorption capacity,
Okay.
these cells have a highly folded apical surface covered in microvilli, tiny finger-like projections that, you guessed it, increase surface area.
It seems like maximized surface area is a recurring theme in cell structure.
It's a fundamental principle in biology.
Right.
Increased surface area means more space for transport proteins,
Okay.
allowing for more efficient reabsorption of those essential molecules.
These specialized cells are incredible examples of how structure dictates function.
They are.
It's amazing how evolution has shaped these cells to perform their tasks so effectively.
It really highlights the power of natural selection to optimize form for specific functions.
This deep dive has been incredibly insightful. I feel like I've gained a much deeper understanding of cell structure. But before we wrap up, is there one final thought-provoking question that our listeners can ponder?
Here's one to consider. Given the incredible diversity of cells from those single-celled bacteria to the complex cells that make up our own bodies, how did such complexity evolve? What other amazing adaptations might we discover as we continue to explore these tiny worlds?
That's a great question. It reminds us that there's still so much we don't know about cells.
Right.
And the journey of discovery is far from over. Thanks for joining us on this deep dive into the fascinating world of cell structure. We hope you've gained a new appreciation for the complexity and beauty of these tiny building blocks of life.